Good day, good afternoon, good evening, wherever you are, ladies and gentlemen. It's been some time since my last talk. A lot has happened, but I'm just gonna talk science today since I didn't get a chance to talk about my last project, which is native fungal plasmids, and uh, we can say actually forest metagenomics as well, but that's maybe that's the third project, after this. So uh, viroids, it's about like fungal plasmids, which I have some experience with, but native plasmids and fungal genome is one thing. Uh, fungal plasmids are another. So it's an area that has never been studied much, I should say. I mean, we barely know the fungal species, in the forest, and I mean, we don't even know the regular DNA sequences of many fungal species, and I am talking about like fungal mitochondria and fungal plasmids and fungal viroids, and it just but it gets crazier. Uh, that's science. That is the ocean, and you have to dive, you know, in the ocean. You have to dive to see, what's in the pelagic zone, and the benthic zone is easy. It's just couple meters, 5 meters, 10 meters. But if you just swim out into the ocean, into the deep, then you get to see the interesting stuff, because out there, nutrients are scarce. I mean, there's not much out there in the ocean. It's just whales, whale poop, and whale carcass, and just deep blue ocean. And back in the day when I was taking classes, I'm just gonna start with that. I took the class called functional genomics. What is genomics? What is functional genomics? I mean, we're trying to make sense of the DNA sequences. Now we have all these cool gadgets. We are sequencing, everything we touch and we see, and just it started with human genome, and it went on with Drosophila, and yeast, and Arabidopsis, and plant species, and etc. Now we have like maybe 5, 10 model organisms, which we know, very good with their genes and their functions, and etc. So even the basic bacteria, like we're talking about 500 genes. I mean, there's this project called minimal genome project, and they just downgraded all the genes to 500, something like that. So, and then when it comes to humans, we have like 25,000, 30,000 genes. Just the code for a protein, that just makes your life possible. Uh, uh, but it doesn't end there. Because when we compile all the data, all the DNA from the ocean, all the DNA from the forest, all the DNA from the soil, all the DNA from the plants and fungi and bacteria, and humans and animals. And we compare these. That's how science works. You just compare and try to make sense of these DNA fragments, and oftentimes we don't know the function of these DNA sequences that we find in nature. You know, we try to compare them to the ones we know already in the database. We can say something. It's called annotation. We can say, okay, this is good for this. This is good for this. But in the jungle of DNA, I tell you that just in the forest of DNA, and this includes space as well, because with the recent findings from the asteroid Bennu, they found building blocks of RNA, DNA, and amino acids. Pretty much they found everything necessary for a cell to divide and grow. Because without these, even for a basic like PCR reaction, you have to include the ATPs and etc. The package comes with the master mix. It had the buffer, the PCR buffer. It has everything, because you need energy to run the PCR reaction, and the stuff comes with it. You pay and not to mention the enzyme. Of course you are paying for the tech polymerase or whatever. Yeah, speaking of polymerases, I will come to the topic. Because it was a mystery, how life started, where did the energy come from, how did it start from RNA, from DNA. So it was a mystery, and with the recent discovery, of the materials found, on Bennu, it was just an asteroid. They grabbed like 120 grams of soil and mixture and sand and whatever, and then they brought it back to Earth. The mission like took many years. I think it started in 2000. It's 16 or 18, I think 16, and it took 2 years to get to the orbit, and just they did some calculations, and they landed after 2 years, and grabbed the materials, and then and brought them to Earth, which is very helpful in understanding life, because the asteroid itself is like same age with the Earth, like 4.5 billion years. So that some rock, with some dust on it, and turns out it has all the elements, for a good DNA polymerization or the RNA polymerization. I'm not saying macromolecules like that. I don't know if they found lipids or anything like that. For a cell, to be functional, of course. Just you may need some membrane and etc. They just started. What they have told us is just maybe 1% of the findings, and then they are actually, I think, willing to donate and give samples to the labs which are interested in the project, which would be good, because you know, because the next asteroid actually, I think they are making calculations, and then like this one is, I think, in 8 years, in 2032 or something. Then now they're like increasing the rate to 3%, of the, you know, of an impact, rate. Is it big? I have no idea. It's just should we do something about it? I don't know. But with this introduction, I mean, it just actually it's a good introduction, because the fungal life, how did it start? 1 billion years ago. I mean, it just the start of the eukaryotic cell, the inclusion of mitochondria into the eukaryotic cell, and the start of the eukaryotic life, the merge of two genomes, which is important. Actually, it is the topic of today's podcast. The integration of genomes, because and then I did that as part of my thesis work too. So it's just you prepare the yeast cells, and then kind of weaken the cell wall, and then with some addition of foreign DNA. I think we used salmon sperm DNA or something. I forgot the ratio, but it was like you have to have enough DNA for the cell to take up the DNA of interest, or the plasmid of interest. So oftentimes you have to incorporate some foreign DNA into the reaction, into the transformation process. That's how you do it. And oftentimes, I think we did it with salmon sperm DNA. Just so you increase the concentration of DNA molecule, and then you just insert like 1 microliter, 2 microliter, whatever, of your choice, So, and then the cell takes up that the procedure. Like, I mean, the transformation of yeast, with plasmids and with DNA, with foreign DNA, it's just tricky, and you have to have a good plasmid and working plasmid, and you have to have the cell ready to take it, and there needs to be enough metabolic pressure to keep the plasmid, and etc. That's a whole different issue. But in terms of diseases, now let's again, let's leave the lab and just go back into the forest again. In nature, I mean, we know that genes, to jump from one organism to another, and usually, these called mobile elements, mobile genetic elements. They exist in bacteria, they exist in fungi, and they exist in plants, and when what we understand from the symbiotic relationships, everybody loves truffles, for example. How does it happen? It takes 5 to 10 years, for a oak tree to take up the mycelium of the truffles, and then start producing, this mycorrhizal fungi. It relies on mostly on oaks, and yes, there are truffles, that grow on pine roots and other roots, and etc. So it's an interesting topic. How does it work? You're talking about like merge of two systems. It's just what is the beneficial element in terms of molecular biology? It just it's easy to say, okay, it grows on it, but how? Is it that easy? What is the mechanism, behind it? So, to understand it, actually, I started, writing another project, on my blog, for myself, to actually improve my own thinking, in the first place. To just write things like a citizen scientist, or like an early scientist, because you have to write down, what you're thinking, and step by step, uh, and looks like it looks like it's an interesting topic, that this fungal native plasmids. It's just and when I mean plasmid is a circular, small, DNA fragment, 5 kilobases, 10 kilobases, not too big, uh, not too big. It's just and what's on it, if it's a native fungal plasmid, what is out there. They are present in fungal tissues. They are present in fungal mitochondria, and from the recent research, what we understand is many diseases rise from uh, the functional mitochondria. I mean, there are a couple genes on them, that is necessary, to catalyze the energy, reactions, in our body. I mean, it's the most important organelle of the cell. I mean, without it, I mean, the cell would starve. You know, the cell would starve, because with glycolysis, the breakdown of glucose, you don't generate much ATP there. I mean, it just check your high school biology books. You don't you don't generate much energy with the glycolysis. I mean, it's just that two ATPs, it just break down the glucose into like pyruvate, or whatever, lactate, or just acetic acid, vinegar. It's just that fermentation, basically, and it just basically, but that is that is not a efficient way of producing energy, though we still need it, sometimes, because your blood runs out of oxygen, during, for example, intense workout, and so our muscle cells and certain cells, actually, they are adapted to low ATP conditions, and then that's why we get tired. We get tired, not because uh, you know uh, there's not enough oxygen. It's just we get tired because we run out of energy. We run out of ATP. The lack of mitochondrial output. You have to have enough supply to run all the genes that is on a mitochondrial genome. On the mitochondrial DNA, there are a number of genes, that needs to run efficiently to give you enough energy, which is required in your brain cells, in your heart cells, muscle cells, bone cells, immune cells, digestive cells, you name it. Without ATP, without energy, it's just the body does not function. It's just like biology 101, basics. I mean, it's just like I said, You can generate some energy with the glycolysis, breakdown of glucose, and that's not enough. That's not enough. So you have to fine tune this energy system, and somehow it happened billions of years ago. I mean, it's just two cells merged merged, not physically, but they also merged their genomes. So we don't know how did it happen, but because there are many research now supporting this theory. Now they are even talking about a specific therapy called mitochondrial therapy, that that you isolate mitochondria and inject into the necessary tissues, healthy mitochondria. And you can Google some stuff that what is good for mitochondria, like Q10 and then the other stuff, the stuff that is good for your metabolism, you know. Even like if it doesn't function properly, now your mitochondria leaks uh, the hydrogen peroxide and all the oxygen species and etc. into the cell, and which makes it toxic and harms DNA, and changes methylation of DNA, causes many maybe unwanted epigenetic disruptions and etc. So, so the inclusion of mitochondria, into the ancient cell, and what happened, during that junction or merger, we don't know. But one thing we know for sure is that mitochondria is still mobile. Now there are even some research showing that there is mitochondria in the blood, free mitochondria swimming in the blood. Can you believe it? Like, remember, I am talking about like mitochondrial isolation, and then we had all this pandemic, and which is everybody became a doctor during the pandemics. We know for sure, and and then now they finally admitted that it was a lab origin, which I told you already. So I thank myself for that. I did a good job on it. So speaking of free mitochondria, so still so we can say it's a mitochondrial theory, it is still running at the human biology, this ancient organism, whatever it is. I swear, because how did I get to this conclusion? Because of my mushroom work. You grow mushrooms, and then one day they're like dying, and the next day they're thriving, and then you try to understand what the heck is going on. Because it's very important, and you have to have fresh tissue, during fungal studies. The mushrooms and the fungal tissue is very susceptible to aging. And what is aging? Aging is maybe 90% related to mitochondria, because without enough energy, you cannot synthesize anything. That includes collagen. That stuff that just and without collagen, what happens? Your space, your face becomes wrinkly and all that, unless you are fat, then you can fill all the blank space, with the fat and the lipids. Still, I mean, you can tell and by looking at wrinkles, in your hands and the feet and elsewhere, and that's the sign of aging. You lose collagen, you lose protein because your cells cannot make it, cannot generate it. Why? Maybe because they are running out of mitochondria. And how do you run out of mitochondria? What goes on it? You know, are there diseases related to this? I think there is one recorded the disease regarding to viroids. I mentioned, but I haven't actually got into the viroid section yet. But so maybe that's the one way to start, because of viruses. The viruses are interesting stuff. They're not living things, but they do have a like a coat, protein coat, which we all know with the spike protein and everything, and the membrane and else, and that's why they said, how do we protect ourselves from COVID? It just, like, show everything to sun, and the sun will light the membrane and the envelope, the viral envelope, and everything. I mean, within 2 years, everybody became a viral expert. That's a good thing. Hope everybody became. Hope everybody learned. What viruses are, but still there is more to learn, because looks like that there's one step, that there's one thing smaller than viroids that can infect us, that can infect plants. As a matter of fact, it causes diseases. In potatoes, I think in apples, viroids, they're called viroids, and they're not viruses. And I think it's and they cause damage in agricultural plants. They cause scar tissue in apples, and malformation in potatoes. Like they become like tubular and elongated morphology because of a viroid. So what's a viroid? That's an interesting topic. It's just so yet smaller, smaller piece of RNA. It has ribozyme on it, oftentimes, which can cleave, RNA in certain locations, and which can form, I think, pinheads, which can fold on itself. RNA is a single strand, not like DNA. But if you're talking about the circular RNA, with the help of this enzyme, called ribozyme, which is very interesting. Ribozyme is a catalytic RNA, which takes us to the asteroid, because we don't know how life started, but we know that certain the nucleotides of the RNA, when they come together, they form ribozyme. Ribozyme can catalytically run and some reactions, I mean, including polymerization. So now they're making experiments. You can like search the Google it. Catalytically active RNA or catalytic RNA. RNA itself. The short strands of RNA can act as an enzyme without proteins. And we know from the cell already. We know the RNA types. We know the transfer RNA that binds to the amino acids. We have this messenger RNA that is the copy of DNA, and we have the ribosomal RNA. Here is the important part. You know, the ribosomal RNA actually, a fragment of it actually is a ribozyme. So you know, it does the work, like stitching all these amino acids together. That's where the magic is. You know, RNA itself acts like a enzyme. You know, because without this magical stuff called proteins, you can find all the amino acids in the space. I mean, if you don't bind them together, it's just good. They're good for nothing. So what is there to bind them? You know, and establish those peptide bonds. Look, we have plenty of time. That asteroid is 4.5 billion years old. Okay, so there's plenty of time till you see the presence of fungi in the oceans. You know, if you have plenty of water, and some rocks, looks like you can get some RNA strands, and from those strands, maybe you can uh, synthesize certain peptides or oligopeptides, or so it's interesting results. Though most of the material they found are like D oriented, not the L, because when you go into the amino acid business, and there's an L-arnitine, L-carnitine, it's just all the amino acids, they start with the L thing, because left and right configuration, and mostly they found the D ones, you know. Oh, why, how, we don't know, but there's for sure, for sure. I mean, if there is then there's enough chance to find the L ones. It's another mystery. Why life started with the L type amino acids. The L-theanine, and just L-histidine, and so on and so forth. So that's another mystery. But looks like, but from all I have talked so far, looks like these viroids, that for some time and for some reason that we have forgotten throughout the history of science, it's coming into the stage. It's coming onto the stage. And guess what? We know nothing about them, and they are present like in plants. And now I think they have found they can be present in fungi. It's called fungal viroids, and they can be present in fungal mitochondria, fungal cytoplasm, but they don't cause disease, but they can transmit. They can transmit these viroids, the fungal tissues. So fungal organisms and fungal infections can be a way to transfer these molecules, ribozymes, and viroids, and uh, and what they found is, the if you study the mitochondrial genome, you, you see many genes, and they are regulated by the nuclear genome. The mitochondria in time, I guess, lost some of the genes, necessary to be completely independent. Most of the mitochondrial, the genes are regulated by the genomics of the nucleus, that is in the cytoplasm. The mitochondria has its own, DNA, circular DNA, and which has certain number of genes on them. But uh, but now it's a whole bunch of different classification. When you try to isolate and classify these mobile elements, you know, and oftentimes I think what they found in common is RNA polymerase to polymerase or to produce itself or to because the replication of viroids are interesting because they to be present they need to be replicated and how they do this is interesting. They don't need an envelope like viruses. Viruses are more complex. It's just and but viroids they don't seem to harm as much as viruses, but still they have some access to the genomic system, and then they can integrate because these ribozymes are the enzymes they can cut RNA. So it's like a ligation. It can make itself circular. It can like synthesize itself like from a single strand, and then it just cuts itself in certain locations, and then becomes circular. It becomes a native plasmid. So on my blog, East Eden Place, blogspot.com. I have listed some publications. I have just started learning about it. Just I because this area is very interesting. The mRNA work. The reverse transcriptase, the conversion of RNA into DNA and DNA into RNA and introns and exons and turns out it's very interesting. There are in mitochondrial genomes they find many genes with introns. Like it doesn't even. Yeast genome doesn't have many genes with introns. There are just a number of them. One or two genes. I studied the yeast genes. They don't have introns. So what is this intron exon thing? It just made life more efficient or more complicated. Maybe it just kick-started the evolution. That's why it is very important. This intron exon thing. The RNA world. We don't know much about it. DNA world is rather it's simple. Yes. So. Even like when you go to a database or open a gene like a mammalian gene, like the promoter region, the upstream region, the stuff upstream of the ATG, the start codon, with the theta box and everything, just the binding, boxes and everything, for the RNA polymerase and etc. For yeast, it's like 500, bases, 1,000 bases, 1 kb. For mammalian systems, it is much longer. Maybe 3,000 or so base pairs to regulate a certain gene. Because when the RNA polymerase starts transcribing, you know, the, the gene, when all the conditions are met. It just binds, the transcriptional activation and all that, good stuff. It binds to the DNA, and it just starts, the translation of DNA into RNA. But then you have this draft, which has, introns and exons, and then this thing called splicing, mRNA splicing. Now we're talking about catalytic mRNA. It needs to be cut, and then these viroids do this. They catalytically change the epigenetics of the system. Very interesting. They work on the mRNA. They just change the splicing system. Maybe they are present in the introns as introns. They just cut themselves out, and then the rest of the the fragments merge and then becomes the messenger RNA. That goes into ribosome to synthesize the protein. But that part is very interesting and important for diseases like SMA, the spinal muscular atrophy. In that disease, there are two genes, SMN1, SMN2, and then the drugs generated so far, either provide gene therapy for SMN1 or enhance the mRNA splicing to get a better protein product. Because I think the intron, I think the intron number 7 doesn't cut properly in that situation. We have the SMN2 gene, but for some reason it is not producing enough SMN protein. And people with that disease, if they are lacking the SMN1 gene and they are dependent on the SMN2, it becomes critical. It becomes vital. So now there are now therapeutic approaches how to fix that. But with the help of viroids, you can regulate these non-functional mRNAs into functional ones. It is possible. That's what I'm trying to say. So in the forest, there are still, many enzymes, RNA-based enzymes, hidden in the soil, that we have no idea. So I try to explain some of the, functions of these, viroids and the RNA molecules, the circular RNA molecules, and what they do in human genome. They can catalyze certain reactions, and they already do in plants and etc. And sometimes cause diseases in the mitochondria too. And what happens is just in time, if we are subjected to these viroids and subviral particles, that just exist in the metagenome of the universe, of the forest, of the soil. They just, like I said, we don't know much about them, but mostly they are RNA based. They catalyze reactions. They work on mRNA, messenger RNA. They can maybe interfere with even ribosomal RNA. They're just small particles, and maybe they just they're just sometimes, oftentimes too small to code a protein. They're just small circular RNA fragments. They don't code, for a protein, but they do. They do some functional work. That was the idea of my project. So what I was trying to do, maybe I would like isolate these, uh, plasmids, native plasmids from fungal tissues, you know, deep in the forest, especially from the symbiotic sections, where the plants meet with fungi, because we know they do. Plants live with fungi and oftentimes make symbiotic connections. And in these zones, just maybe use some restriction enzymes, and then try to get some DNA, plasmid DNA. It's a different molecular protocol than the genomic DNA because they are circular. You can uh, you can use different approach to isolate and extract them. Maybe we can use some RT-PCR or other work, but I just want it to be simple. That just simple enough to like extract mitochondrial DNA, plasmid DNA from fungi, from those interaction sections, uh, uh, and cut them with certain restriction enzymes, which has certain patterns on it, and it looks like they are present already, on the hairpin sections of these viroids. That you can you can identify these motifs. So would it be helpful to sequence them and just checking the clock, and I'm into 1 hour almost for the 7th minute, so I'm just trying to finish up and summarize. Uh, like I said, in the metagenomics of the forest, of the fungal kingdom, this is important, even like for analysis of species, like it's called DNA barcoding. Yes, mitochondrial DNA is very useful. It provides most of the time 96-97% resolution to identify a species, but yet not necessary. Oftentimes you have to use the ribosomal 16S ribosomal sequences and etc. to complete to get a full picture. Why? Because most of the viroids, they integrate themselves into the genomic DNA. In my blog, you will see publications regarding this. From viroid to mitochondrial DNA, and from mitochondrial DNA into genomic DNA. Mitochondrial DNA is something else. It is more complex. Trust me. It's just and we have to understand mitochondria very well. Very 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 well, because it looks like a exchange hub. Between the viroid world, the RNA world out there that we don't know nothing about, with the genomic DNA that is established in the living things. So mitochondria is the adapter, to say, So as an adapter, oftentimes it can malfunction. so that's the idea. Very important. Many diseases are now, based on mitochondria, because it is susceptible to these viroids and susceptible to mutations and deletions, and so mitochondrial health is very important. I'm just gonna give you an example here. See, this is my I'll show you. This is actually uh, uh, my mycelium growth from agar. I transplanted agar, and this section, as you see, there's this line, and it's not so white in the center. See, yeah, this section. See, there is this line like a like a like it's drawn like with chalk. That's the intersection. That's the border between mold and the oyster mushroom mycelium. So what's happening in that border, we don't know. So based on my experience, we don't know. For example, we know that many molds carry these viroids that they get from plants, because most of the time molds grow on plants, and they carry these viroids because viroids mostly live on plants and cause agricultural damage and many other things we don't know. So and what was this? This was like oat. It is it's important. And to my knowledge, I sterilized this to 2.5 hours in the autoclave, in the pressure cooker. But still, that if you are unable to kill a mold, one single one, just get with a viroid in it, that it got from a plant. Remember, this is what? This is oat. Stuff came from field. Just the cosmos. It came from field. And this intersection is all about, the 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 field of both worlds. Molds are something else. They grow fast. They're most of the time pathogenic. They grow on the plants and etc. So see, the mycelium kind of encircled it. It is, hopefully in time it will close. Eventually, win over the mold. If it doesn't have anything dangerous, but it all depends on the mitochondria. If the mycelium, if your mushrooms are healthy, and if you don't contaminate them, with other molds and stuff. That's why molds are bad for your grow houses. If in your grow houses, if you don't sterilize your substrate very well, it will cause epigenetic changes. You don't know what's in your mold. Most of the time, you will see these viroids. That's why we need to understand and study, what's out there, what type of molds carry these viroids, and what they do, how they cause harm. Do they cause harm? And in those symbiotic relations, how do, like, can we grow truffles in acacia trees? Why stick to oak? Acacia grows in dry areas very well. Or how does it work? How do you get truffles into the roots of a plant? How do you establish that? How do you establish that connection? We know with yeast that see they don't form the clamp connection. In yeast, this thing called clamp connection, when two different strains of yeast or mycelium, they meet, they exchange, you know, subcellular material, they merge the hyphae, they merge the system if they're close enough, you know. Even to go back to basics, you know, what's an egg? What's a sperm? We know sperm doesn't have any mitochondria. An egg has lots of mitochondria. We have this concept of mitochondrial Eve. When we go back to the beginning, maybe the first cell with the healthy mitochondria became the first egg. You know, you know, because we have the answers. And in yeast we see this. You know, in yeast there is no like egg and sperm. It's there are two, you know, compatible cell types. You know, that is, you know, meiotically acting like, you know, egg and sperm, but it's called A and alpha. You know, and when they are compatible, they make the, you know, so-called zygote. You know, so it becomes diploid. You know, yeast cells, they can grow as haploid cells. You know, you know, when there is this haploid cell A and the other haploid type alpha, you know, when they meet, it becomes the diploid cell, and then and then that diploid will go through meiosis. A haploid cell won't go through meiosis. Then it doesn't have enough chromosomes. So this mitochondria stuff is really interesting, and it has implications for human life, for diseases, for agriculture, for many things. It's just a start, you know. It's just like I said, these are not viruses, but yet smaller than viruses, and then just like the immune system. We don't even know our immune system is adapted to fight these things. You know, the viroids, just what they do, what they don't. We have to learn. We have to learn because, you know, with the pandemics, you know, it's just we learned, you know, a lot about viruses and their structure, and these are a lot you know, simpler in structure, and in function wise, it could be vice versa. They can have very functional, you know, impact on human genome and plant genomes and etc. So, like I said, I'm just learning about this viroid stuff. You know, the introns and exons and, you know, and ribozyme. You know, the term. I just, you know, read it. And ribozyme, catalytic RNAs, and, you know, just and what RNA can do, and, you know, what's RNA, and RNA-based therapies, and etc. Ribozyme-based therapies, mitochondrial, you know, therapies, the mitochondrial health. So it's just a whole bunch of, you know, because, you know, that's what happens when you grow mushrooms. I mean, it just, they get old very quickly. I mean, the aging. If you want to study aging, you study mushrooms because they age very fast. They rely on, you know, regeneration. You know, the thing we see on forest, it's called fairy ring. You know, it gets bigger and bigger. You know, the mycelial mass, you know, grows into a ring. They call it fairy rings, and every year it freshens itself and it gets bigger. It becomes a bigger ring in the forest, and you see all these mushrooms growing in a ring pattern. You know, because just That's what mushrooms do. Maybe they incorporate the mitochondria from the old ones and regenerate cells and it's all about regeneration. Like I said, mitochondria is a mobile organelle, I should say. That's just to summarize it. It's a mobile organelle, including our body, human body. There are reports that we have swimming mitochondria in the blood. It just in cases of disease and etc. This becomes very important. Because we're talking about all these blood clots and etc. This just became a thing with the pandemic, and it's still the case. A lot of people suffer from clogged arteries and etc. What's in your blood? See, it's not only platelets and red blood cells and erythrocytes. There's mitochondria too. So, and God knows what's happening. Where these mitochondria go? How do they merge into other cells? It's just only area. Ladies and gentlemen, thanks for listening me. So I will continue reading and learning about this topic. Like I said, this is based on my work and experience, because I'm trying to grow mushrooms and I try to better my cultures and but just sometimes just every time I get a piece of fungal tissue, healthy fungal tissue, I stick it into the sawdust bin I have. So I'm providing a healthy mitochondrial transplantation into existing tissues, because if they belong to the same species, that the mitochondria will migrate from one tissue to another. It's like an medical aid, supplement. So even for plants, it's important. And if there is any a lot of people now pruning, the gardens and etc. Please sterilize those equipment very well, because from an infected plant you can contaminate many other plants with an infected utensil. So just make sure sterilize those scissors and whatever knives and etc. you are using to prune your trees in your garden, so that you don't contaminate your plants, and don't blame the bugs and everything else, and some maybe it can be even carried with bugs and such. We don't know exactly how this thing works, but it's an important topic. Okay, ladies and gentlemen, have a good evening, good night. Thanks for watching.